So today we're going to be presenting on Chagas disease, also known as American trypanosomiasis. And I'm Andreas.
I'm Tavia.
And I'm Tiffany. So we're going to begin by talking about the life history of Chagas disease. Then we'll move on to the ecology and evolution of it. And then we're going to identify a knowledge gap and propose an original experiment.
I'm going to start with the life history of Chagas disease. First, I want to start by talking about the pathogen that causes Chagas disease. The pathogen that causes Chagas disease is the protozoan parasite called Trypanosoma cruzi. And this parasite kills 30,000 people a year, most of which being women and children, unfortunately. And below, you can see what the parasite looks like under a microscope. They resemble an S shape or a C shape under a microscope. And the circles are blood cells you can see. Next, I want to talk about the intermediate and definitive hosts. The intermediate host is the triatomine bug or the kissing bug, which is identified most commonly by the specific patterns on its back slash shell. And the definitive host are mammals and humans. The signs and symptoms present themselves most commonly with a swollen eye, which is one of the most common signs that somebody has contracted Chagas disease. And then you can see in the bloodstream, once again, where the parasite would live. Chagas disease is most commonly transmitted through the feces of the triatomine bug or the kissing bug. And it follows a specific chain of infection. First, the bug will bite the human, leaving its feces behind. It's important to note that the infection is contracted through the parasite, which is found in the bug's feces. The feces that it leaves behind, the parasite then leaves the feces and enters the human host through the mucous membranes or breaks in the skin. And then finally, the human is infected. Other ways that humans can be infected with Chagas disease are orally through things like contaminated food, vector transmission through the bug, organ transplantation, blood transfusion, congenital, which is from unborn baby from the mother, work accidents in the laboratory. This is going to be specifically dealing with the parasite itself or the disease, and then finally sexually. The acute phase or the early phase of Chagas disease is most commonly asymptomatic or mild. The most common signs and symptoms include things like fever, body aches, fatigue, and or swelling at the site of the infection. The chronic phase of Chagas disease presents itself with cardiac and gastrointestinal symptoms. The cardiac symptoms most commonly experienced are arrhythmias, heart failure, or sudden cardiac death. And the gastrointestinal symptoms most commonly experienced are megaesophagus, which is the swelling of the esophagus, which the esophagus is the pipe that leads from your mouth to your stomach. And then the megacolon is the swelling of your colon. The severe complications from Chagas disease are all continuations of cardiac or gastrointestinal diseases. First, we have myocarditis, which is inflammation of the middle layer of the heart. Then we have meningoencephalitis, which is inflammation of the brain and surrounding tissues. And finally, we have hepatosplenomegaly, which is inflammation of the liver.
And now we're going to go into the ecology and evolution of Chagas disease. So Chagas disease traces its origins back to 7 to 10 million years ago from a bat trypanosome. And evidence also suggests that indigenous populations in South America were affected by T. cruzi as early as 9,000 years ago. And it likely originated specifically in the Andean region, spreading as settlements expanded and deforestation increased. And there are also historical accounts from the 16th century that describe Chagas-like symptoms affecting both indigenous peoples and also the conquistadors. And now transitioning to the modern era, we can trace the identification of T. cruzi and triatomine bugs to as early as the 20th century. And as mentioned, environmental changes such as deforestation really facilitated this spread of Chagas, creating modern challenges such as outbreaks in previously unaffected areas and also insecticide resistance. So some environmental factors that affect its prevalence. So its distribution spans from Mexico to northern Peru, also including Colombia. And this is significant because we found this case study about the environmental determinants of Chagas. It was specifically in Colombia. And it was found that it occupies various ecological zones, going from sylvatic to domestic habitats. And it also has a large epidemiological importance as it poses a huge risk, specifically in the central and oriental regions of Colombia. And its species distributions and abundance are affected by various environmental factors. So some of the environmental factors that were identified were temperature, precipitation, vegetation type, and also land use. And analytical tools like GIS, remote sensing, and niche modeling were used to understand these relationships, along with predictive modeling as well, using logistic regressions and ecological niche modeling to predict these distributions. And some of the variables that were observed were maximum vegetation index, minimum land surface temperature, and also digital elevation as predictors. And now understanding these ecological interactions is crucial for effective disease control and also for our proposal for our experiments. So as Tavia mentioned, the stercocarrier mode of transmission is very unique, but also very effective, which contributes to the spread of the disease. And it also has a very broad host range, being able to infect a wide range of vertebrate species, really complicating disease control efforts and underscoring the importance of understanding host vector interactions. And also its longevity and nymphal hematology leads to gradual increases in infection rates throughout their lifespan. And for reference, its lifespan is typically 1 order of magnitude longer than that of other vectors like mosquitoes or flies, really contributing to this difficulty to control Chagas.
Next, we're going to go into a knowledge gap and propose an original experiment related to Chagas disease. So our question of active research we're going to be addressing is what are the long-term effects of habitat fragmentation on the phenotype and genotype of triatomine bug populations in the United States specifically? This picture down here is of a continuous forest whose habitat was fragmented because of a road. So what is known about habitat fragmentation in triatomine bug populations? So habitat fragmentation is basically when a large expanse of an original habitat gets divided into separate smaller patches due to natural or man-made causes, natural through wildfires or possibly volcanic eruptions, but usually it's due to man-made infrastructures. We know that triatomine bugs prefer rural areas, but recently research has found that they are moving to peri-urban and urban areas at an increasing rate. This is due to factors like climate change, which is causing these triatomine bugs to move more north, as we can see in this map of the United States of the bottom states are all gray, meaning they have had triatomine bug spottings. And then this is also due to habitat fragmentation with humans inflicting their infrastructures onto these triatomine bug habitats, leading them to search for their food resources elsewhere and have to come into more increased contact with humans, leading to more cases of Chagas disease. We also know that bugs, when they're separated with one another, cannot breed, which leads to genetic isolation and reduced gene flow. So what is unknown about this topic? Research studies have only covered the short-term effects of habitat fragmentation on triatomine bugs, and this is usually in South American countries, not the US. So as these bugs are moving northward, I believe we should study them as we are inflicting on their habitats. And then we're also going to look at the coevolution effect hypothesis, which was recently released in 2019, so it has limited scientific backing as of now. But by investigating this, we can make conclusions about local adaptation, loss of genetic diversity, and the effect of environmental stressors on these triatomine bug populations. So this graphic here is just showing three separate parts of a habitat that used to be one. And it shows that in the coevolution effect hypothesis, these species are now all isolated from each other, so their gene pools are in three separate parts, so their evolution trajectories are completely different. So in our original research experiment, our study objective would be to determine the long-term effects on phenotype and genotype of U.S. triatamine bugs based on fragmented habitats and intact habitats, and fragmented sections. So our hypothesis would be that triatamine bugs from the separately fragmented areas will present different phenotypes and genotypes over a long period of time, as well as it will look different from the intact habitat bugs. And this picture down here is from a research study where they compared the heads of southern Andean triatamine bugs to northern Andean triatamine bugs. And we can see from the different habitats they're in, the top one had a lot of nests in its environment, so it adapted and had a smaller head to fit into these nests, versus the northern one lived in the wet forest palms and is a lot bigger head. So our methods of this original research experiment would to be locating triatamine bug areas in the US with differing levels of habitat fragmentation. What that means is we'll take some places with very split habitats, then medium level split habitats, and then very low evidence of fragmentation, and then an area that has no fragmentation at all with triatamine bugs in it. Then we would do monthly bug sampling to look at the population characteristics and assess how it's changed. In the data analysis and results portion, we're going to genetically analyze the DNA of the triatamine bugs and look at the phenotypic traits for each bug group over decades. We're going to use machines like this DNA sequencer to really assess the changes. And with that, we can gauge the trends as well as create models that will help us inform future public health decisions related to Chagas disease. And that is our original research experiment. This is our bibliography. So thank you so much for listening to our presentation.